Hey Beaks, today we're going to start cellular respiration. So I hope you already did your part and you read the introductory modules and got your head wrapped around what this whole chapter is about. And we're going to do a little bit more of that today. And then, like I said, eventually this stuff just doesn't click for all students right away. But usually if you draw it out with me and you really see the whole picture, then all of a sudden it's like, oh, okay, I could do this. Cellular respiration isn't too bad. So today is just some background information. Let's take a look. So we're in the mitochondria for the most part this chapter, but not the entire time. So cells tap energy from electrons falling from organic fuels to oxygen. Okay, so I'll explain what that means by the end of this unit, but basically you're going to take an electron from one type of bond and move it to another type of bond. And if you understand your chemistry pretty well, this chapter will make a lot more sense to you. For example, do you remember that hydrocarbons we talked about, which is carbon, hydrogen, right? Those are really good fuels, okay? And if you remember things about like electronegativity and polarity, remember that carbon, he's not too much of a bully. So when he's sharing electrons covalently bonded with hydrogen, okay, those electrons are out there flying around him. Whereas oxygen is kind of a bully, right? And he's electronegative. So he's going to be pulling on those electrons, keeping them closer, okay, to himself. And if you remember, there's more potential energy available to do work in an electron that's farther from the nucleus than one that's closer to the nucleus, right? So we're going to end up taking a carbon-hydrogen bond this chapter, taking the electron from there and putting it into an oxygen-hydrogen bond where it's kept closer because oxygen's a bully. That little change in energy, okay, changing in the electron orbitals, that's how we get our ATP. That's how we're able to function, okay, and get our energy for the most part. So the energy available in a cell is found in the arrangement of electrons that form chemical bonds in organic molecules like glucose. Remember, that's potential energy. And your potential energy can either be on the location or the structure. So here we're talking about the structure, right? Glucose, C6H12O6, lots of electrons in there, carbon-hydrogen bonds that we're going to be after to manipulate. And we're going to make good use of those electrons or the potential energy that they're storing. So electrons are going to be transferred to oxygen as the carbon-hydrogen bonds of glucose are broken and the hydrogen-oxygen bonds of water form. So actually you're going to break down glucose this chapter and you're going to end up making water. As electrons fall to oxygen, they lose potential energy. Okay, we control this energy release and slow it down, kind of like going down a staircase, and thus are able to store it in ATP. And that's a very important concept this chapter. You don't just release all the energy of your food at once. That would be dangerous. If we had time to do it this year, do you remember burning food when we did our calorimetry lab? Okay, if you did this with me, you know that we had one cheese curl. And when we lit that cheese curl on fire, you got this huge flame, right? Well, could you imagine if you released all that energy all at once in your body? Can't do that, right? So the whole point of this chapter is to think of it as a series of slow down steps, right? Like a slinky going step by step by step down a staircase. And every time the electron falls, if you want to think of it that way, you release a bit of energy. And that way it's not a ton of energy, right? This huge exergonic reaction. You're talking about little bits of energy coming out that we can hopefully harness along the way. Of course, we're not going to do a great job of that. Right? Remember the second law of thermodynamics. Well, the first law says anytime you have energy, you can transform or transfer it, but not create or destroy it. Right? That's the law of conservation of energy. But the second law says, all right, go ahead. You're going to play around with energy. You're going to transform or transfer it. You're going to increase chaos. You're going to increase heat. And that, for the most part, is why we are 98.6 or thereabouts degrees. Right? Because we're breaking down our food slowly, and every step we break a little bit down, entropy, right? We release a little bit of heat. Okay, so there's two words this chapter that you really have to understand, and you have to be able to talk about them very well and just feel comfortable with them, and those are oxidation and reduction. Okay, so oxidation, reduction, or redox reactions are just the movement of electrons from one molecule to another. All right. So in a redox reaction, the loss of electrons from one substance is called oxidation, and the addition is reduction. Okay, and that should make sense to you if you think about the reduction, right? You're adding electrons, which are negative, thus reducing the charge. All right, now again, some of you right now might already be like, wait, what is going on here? This chapter is already like, bleh. It'll make sense at the end, I promise, okay? Because right now you're at the beginnings of a pretty tough chapter, and depending on your background, a lot of you, if you're sophomores right now, you didn't have chemistry yet, you didn't have physics yet, so this stuff's a little bit tricky for you, okay? Or if you're freshmen as well. So here's a little jingle to help you remember oxidation and reduction. There's two of them, actually. You can think oil rig, that's the one I like, or LEOGR. So oil rig, oxidation is loss, reduction is gain, okay, of electrons. So if you're going to oxidize something, like we're going to do to glucose this chapter, that means we're going to take the electrons from glucose. Oxidation is loss. Now, if you're going to reduce something, they're getting the electrons, right? Reduction is gain. Okay, so you'll see what happens with oxygen this chapter. We're going to give it some electrons, so that would be reduced. Or you can think of it as LEOGR, okay? So Leo, lion, right? And GER. So LEOGR, lose electrons is oxidation, gain electrons reduction. Okay, so no matter what little jingle you use there to keep them in your head, these two words have to be very, very organic for you to say, right? You have to be able to understand oxidation and reduction reactions to the extent that we'll be covering them in this chapter. Okay. So here's what actually is happening this chapter. You'll always need a donor and a receptor for redox reactions, okay? And this equation will be very important for you to wrap your head around. That is the equation for cellular respiration, all right? Now, some of you might already be wondering, wait, don't we eat food besides glucose and things like that? Yeah. You can, do oxidative phosphorylation with almost any type of fuel, but this is the one that we have completely mapped out, and we're not going to get into every little, step in here, these make-ready reactions, but glucose is what you normally will break down your food into, and you'll have this blood glucose level, right, that you can use for energy. So that's why we talk about glucose. But here we go. Glucose, you eat this in your diet, and you're going to rip the electrons out eventually, and that's called oxidation. Oh, and you're left with CO2. Now, you also breathe oxygen, right? This is why you're like a furnace. Think about it. A furnace needs oxygen, right? Your car engine needs oxygen. Anytime you have a fire going, you need a candle, you put a lid over it, it'll go out, right? Because flames need oxygen. So for you to burn your food, you're going to need oxygen. That oxygen is going to get reduced, okay? And remember what reduction means. This is when you gain electrons, and look at that, you become water. Okay, and the whole point of this is to get energy in our case in the form of ATP, but of course we lose a ton as heat as well. Okay, so it's kind of weird. For the first time this chapter, you might actually understand that some of the things that you've been thinking your whole life are a little bit wrong. For example, why is oxygen so important? You're going to see this chapter. All oxygen really does for you carry it around in your blood so it gets to all your tissues, is really just, it's an electron acceptor to make water. Right? Oxygen is actually a very dangerous caustic gas. As a matter of fact, when photosynthesis first started on planet Earth and oxygen started becoming more prevalent in the atmosphere, it was a mass extinction event. Right? Oxygen is an electronegative bully. It's a dangerous gas, and it killed a whole lot of organisms. So we've now evolved, our species have evolved to need it, right? And so do many animals out there, but we need this oxygen. However, it probably leads to us aging. There's these things you'll learn about later called free radicals and things like that build up. And it's, wow, we need antioxidants. We need to get rid of some of this junk. Oxygen's a caustic gas, okay? And the other thing that you might not have known is every time you exhale, you're breathing out CO2. And where did that CO2 come from? Your food, right? How weird is that? So CO2 that you breathe out is actually, thanks chicken nugget. Because that chicken nugget, a lot of the carbon that's left over and some oxygen becomes CO2. So you're actually breathing out the food that you ate. So pretty cool stuff, and we'll get more into this. And one of the neatest things about this is the next chapter that we get into is photosynthesis, and you're going to see that plants do the exact opposite of us, right? Plants will say, oh, you're giving up CO2? Sure, I'll take that waste product. You're giving up water? Sure, I'll take that waste product. Okay, now again, we need water as well. We wouldn't give it all up, but just a couple molecules here are the products of this reaction. So plants are like, yeah, give me your CO2 and H2O. You know what I'll do? You think these are low energy crap? You took all the energy out to make ATP? Well, I'm going to use the sunlight to re-energize it, boom, and make sugar for you. And you know what I'll give you back? I'll give you not only the sugar, but the oxygen. So plants take in our products and they use them as their reactants for photosynthesis. They do photosynthesis, and guess what their products are? Our reactants for respiration, glucose and oxygen. Pretty cool. So we're in this neat cycle with plants. Okay. Now, how do you actually get the electrons out of glucose? Okay, or out of the intermediates that are about to come? Well, there's a family of enzymes called dehydrogenases. Okay, and they work with a coenzyme. And if you remember what a coenzyme is, that's a little organic helper, okay, for the enzyme. And his name is nicotinic adenine dinucleotide, or what we're just going to call is NAD+. Okay, and he's an electron carrier. So basically, this dehydrogenase enzyme, we're going to draw it out, will fit glucose in, and he's going to strip some hydrogen, okay, off of the glucose. He's going to allow NAD+, which helps him work. He's going to give the electrons to him, and that'll release some protons, H pluses, then once the electrons are gone. Okay, so that's kind of the whole point of how we're going to get these electrons. And again, right now, you're probably thinking, what in the world? We'll get there. Okay, I know it's tricky right now. Okay. So to go a little deeper right now, and again, we'll draw this out soon. Dehydrogenase strips away hydrogen atoms. Okay, and then the NAD+ organic molecule made from the vitamin niacin picks up and carries the released electrons. That's the whole point of redox. We're going to take the electrons, we're going to put them somewhere else, right? So for example, let's just say that this is one of our remnants left of glucose. All right, we've been ripping glucose apart, and here's what's left of it for right now. We're going to do oxidation on this. We're going to go in here and we're going to grab some electrons out of here. All right, but how do we get the electrons from these bonds right here, from carbon, hydrogen, in this case, different kinds of bonds? Well, we're going to use dehydrogenase to strip those electrons, and that's going to rip off the hydrogens, and then we're going to have this coenzyme, NAD+, is going to say, oh, cool, let me have those electrons, okay, and then I'll, you know, the proton, one of them gets carried with it, actually, that's why it becomes NADH, and one of them is loose, which is also going to be important. That's reduction, right? You took the electrons from the bonds of what's left of glucose, and you added them, okay, so that's oxidation. You added them or reduced NAD+ to become NADH. And that charge should make sense, right? Because you had NAD+ and you added a negative, okay, so now you'd be at NAD, then you added another negative, so technically you'd be at NAD-, but then you added a little proton, which is an H+, so now you're at NADH. So the charge works out, and plus you release a little proton here as well. All right. So the NAD+ picks up 2 electrons, one neutralizes the positive charge, reducing the molecule to NADH, and one is released. So NADH, what is its goal? This is known as an electron carrier. Okay, and it carries the electrons to the first step in the energy staircase known as the electron transport chain. So that's kind of like a funny way to say it. Eventually, when you guys see this entire system and how it works, you'll be thinking, energy staircase, what are they talking about, right? You're going to know it so well, and you're going to be into all the complexities of it. But for right now, just think of these electrons going to a staircase. You're going to drop these high energy electrons that were from these really strong covalent bonds to this top of a staircase, and think of it as a slinky going down. You're going to go down the staircase and lose that potential energy. Okay, we're going to harness that energy. Some of it's gone as heat, but the rest of it will harness to make ATP. So this is going to be located, do you remember the cristae, the inner mitochondrial membrane, all those twists and folds? The electron transport chain is located within the membrane, okay? And there's going to be a whole bunch of electron carriers in there. And in the series of redox reactions, you're going to deliver here. So here comes my NADH. We just got the electrons, right? We're going to deliver them to the chain, and thus that means he's now getting oxidized, and the chain is getting reduced. Right? So now you're back to NAD+, and you released a proton, right? And he's going to say, oh, okay, I'll go pick up more electrons for you, Mr. Electron Transport Chain, right? But now what happens to the electrons? They get passed along, and every time they get passed, you harness a little bit of energy to make ATP. And it's certainly more complex than that, Okay, as a matter of fact, by the end of our drawings, you should look at this picture and be like, no, thank you, I'm not going to look at that anymore, because it won't make much sense to you once you really understand the chapter. And then what happens at the end of the chain? Well, you always need a way to get rid of your low energy electrons, right? So now who are you going to reduce? Well, you're going to reduce some oxygen, which is pretty cool, right? That's why we breathe in oxygen, is so we have somebody at the bottom of the chain to take it in. Now, oxygen's a diatomic molecule, meaning there's 2 atoms to it, so they write 1/2 here just to help you understand that, okay, half of the oxygen molecule, okay, or 1O, if you want to think of it that way, will take in 2 hydrogen ions, so now that's H2O. Well, that's not just water without electrons. Oh, here come the electrons, right? So 1/2 of an O2 molecule is going to become water, and the other 1/2 of an O2 molecule is going to become water, right? So oxygen serves as a greedy little guy at the end of our electron transport chain to take our electrons away. Comprende. So what chemical characteristics of the element oxygen accounts for its function in cellular respiration? Think about that. And it's that it's so electronegative, right? It can pull electrons from other elements, right? Because it really loves those electrons. Very electronegative atom. Okay. And just a little bit of a background today. So before we draw our pictures together, an overview. Cellular respiration occurs in 3 main stages. All right. So the first stage is going to be glycolysis that we talk about. And this occurs in the cytoplasm. It's going to break sugar into 2 molecules of what we call pyruvate. A tiny amount, 2 actually, ATP are generated, and electrons are going to be transported to the electron transport chain. Okay, and glycolysis, because it happens out in the cytoplasm, anything can do this. Okay, even prokaryotes, right? Because they don't have these big complex organelles like we do. Matter of fact, this used to be a prokaryote probably, right? Or mitochondria. So glycolysis, you start off with your glucose in the cytoplasm, and we'll see how you bust it in half basically and do some more stuff. And you're going to end up with 2 pyruvate. So you didn't do a whole lot out here. Okay, you did get 2 net ATP, and then you end up with some electrons that are going to go to the mitochondrion. Okay, but where's the energy of your food is really still here in pyruvate. You didn't do a whole lot, but I'll tell you what, pyruvate is welcome into your powerhouse, right? Pyruvate can come into the mitochondrion. So glycolysis is a hugely important step for us to do cellular respiration. The next thing that we're going to talk about is the citric acid cycle, or the Krebs cycle. And what this is going to do is it's going to take your pyruvate and it's going to oxidize it. Okay, so it's going to, first of all, pyruvate here, you have to groom it a little bit and get ready for this cycle. Okay, and when you do, yeah, you're going to get a little bit of ATP, but you started off with C6H12O6. By the time you're done grooming your pyruvate and going through this cycle, oh my goodness, look at this. You've ripped it to shreds. You've really oxidized the heck out of this molecule, and you stole so many electrons from it that you're really not going to have much left. Okay, so when we get into, this is kind of like the furnace part, right? When we get into your mitochondria, you're going to see how we really just shredded our glucose, okay, or what's left of it. And then finally, oxidative phosphorylation includes the electron transport chain and something that they call chemiosmosis. And this is where you're going to get a whole bunch of ATP. Okay, and this is why we love NADH carrying its electrons in, right? And I'll explain this guy too. He's basically the same as NADH, but he comes into the Krebs cycle, FAD. So I'll tell you about him in a little bit. So what's the whole point here? If you really want to think of it this way, you went through all of this really just so you can get electrons. And what do you do with those electrons? You put them at the top of your staircase, and as they fall down the staircase, those once high energy electrons are going to lose their energy, and oxygen will pick them up at the end to make water, but you're going to be able to make a whole bunch of ATP. Okay, about 28, and that gives us about 32 for the whole process, right? Okay, so 28 here, 2 here, 2 here, about 32, but that number can vary a tiny bit. All right. All right, so almost done here. The majority of ATP production in respiration is generated by oxidative phosphorylation, which uses the energy of electrons to phosphorylate ADP to ATP. So here we go. We're going back to chapter 5 a little bit here. So remember, adenosine triphosphate has 3 phosphate groups. And if you remember, I told you that the bonds of those phosphates are kind of like compressed springs, right? They're on there. They're not super stable. They want to pop off, right? And if they do, they can go and add their energy to a different system, okay, or a different molecule or whatever. We're doing the opposite here. We're taking ADP, adenosine diphosphate, and we're phosphorylating that. We're taking a high energy phosphorus here, and where are we going to get the energy from? You'll see in a second, we're going to use this concentration gradient. And we're taking the energy available and adding the P to the ADP. So we're actually making ATP from our food, okay, or from the energy that is the result of our food, all right? So how do we get this energy? Well, the electron transport chain passes electrons. It pumps hydrogen ions or protons across the cristae, creating a concentration gradient, okay? And it's kind of weird right now to think about that, but eventually you're going to see all these pumps pumping up all these protons into a little tiny area called the intermembrane space, and there's going to be so much of it up there. It's like a waterfall, and it just wants to fall down. There's so much potential energy there, and as it falls back down, we're going to harness that energy, okay? So that's coming up too in our drawing, so don't worry if you don't quite get that yet. And then in chemiosmosis, that's what I'm talking about here. The potential energy created by the gradient is used to make ATP by the diffusion of these hydrogen ions through ATP synthases, which are these big protein complexes in the cristae that make ATP. Pretty cool. All right. So comprende. Of glycolysis, the citric acid cycle, and oxidative phosphorylation, which one uses oxygen to extract the energy from organic compounds? All right. Now, this is a tricky question, actually, because glycolysis just happens out here in the cytoplasm. Whoa, there, I just gave you the answer. You don't really need oxygen for this, right? And do you need oxygen for the citric acid cycle or the grooming or anything like that? Not really, right? But you do need it for this because oxygen picks up the electrons at the end of the chain. Okay, so that's why it's oxidative phosphorylation. Okay, there you can see the oxygen there. Now, having said that though, technically, indirectly, this shuts down, you're going to learn, without oxygen. So if you don't have oxygen, the citric acid cycle is no more. Okay? And if you don't have enough oxygen, glycolysis kind of, takes on a whole different form as well. We're going to do something called fermentation out here. So without oxygen, this whole process kind of gets goofy. But yes, if the question said maybe more directly, which one directly needs oxygen to work? Certainly oxidative phosphorylation using an electron transport chain and chemiosmosis. All right. So I know that's goofy, and I know that's the very beginning of a complex concept to wrap your head around, which is cellular respiration. So my advice is this. Take it with a grain of salt for right now. Try your best to get down some vocabulary and get the big picture idea. And once we draw out cellular respiration together, I think if you came back and watched this video, you'd say, oh, that's easy. I get it now. Okay, but until you see that, it might be a little tricky for you. So good luck, and we'll talk with you again soon. Take care.